Hello again, nice to see you. I hope you're doing good. Today I want to actually look into something very important for those writing the West African examination that is forthcoming, and I'm going to be narrowing my discussion on biology practicals. And mostly when you're looking at science subjects, these science subjects where you actually get more marks for any exam is mostly the practicals and the theory or the essay. And I want to actually do a little rundown of some of the things I believe will be possible questions that will be coming up concerning the specimen or the specimen that are actually provided or will be provided during the Waec practical biology exam. And if you're ready, I would like you to get your pen, your books, and also click on this channel and subscribe with us, okay? First of all, I would like to list the specimen that is actually or that will be needed for the biology practical. I'll start with specimen A. Specimen A is the gills of the fish. Now, permit me, I didn't come with the gills of the fish as to show you, but definitely we will be looking at diagrams of the gills of the fish. In biology practicals, you will not be asked to do any dissections, which I believe I've not seen it. You won't be asked to do any dissections. You won't be asked to carry out one or two practicals as it relates to this. If it had to do with food tests, then it is possible for us to have looked at one or two things to actually put together in terms of a reaction and then come up with a positive or actually come up with an inference of what we're actually looking at. But in considering the specimen that was provided or that will be provided for these upcoming biology practical 2024 Waec, I believe that what you should be taking note of is the pictures, being able to identify these specimen when it is provided for you during the exam. So permit me to list them out. In subsequent time when we start talking about each of the specimen, you will see the diagrams of these specimen. So specimen A, like I said, is the gills of a fish. Gills of a fish. No specific fish, but just the gills of the fish. It's not the gills of tilapia fish or any other kind of fish, mackerel or all that, just the gills of a fish. Specimen B is whole liver from domestic fowl with gallbladder attached in a petri dish. So it must be produced, it must be carried on a petri dish. And then specimen C is the lungs from still the same domestic fowl, but it should be kept on a white tile, The next one is, lungs from a domestic fowl on a white tile. And then we also have specimen D, which is the whole gizzard from a domestic fowl in a petri dish. Specimen E is whole intestine from a domestic fowl spread in a tray. Specimen F is the ginger rhizome. Then we also have specimen G, and that should be onion bulb. And then we have specimen H, which is leaf of bryophyllum with adventitious roots attached to it, okay? The leaf of bryophyllum with adventitious roots. We also have specimen J, which is cassava stem, and specimen K, which is cassava tuber. And also we have specimen K, or that should be L or so. We have thoracic vertebra. Specimen M, we have humerus bone, that's the humerus bone. Specimen N, we have the mango seed, and specimen P, we have the femur bone, And also we have other specimen. We have specimen, I think Q, which is the lumbar vertebra. And then we also have specimen R, which is the orange seed. Okay, I believe I also mentioned the mango seed, that's specimen N. And all these are the specimens that are to be provided during your Waec practical exam. But before we go ahead, it is actually important for you to understand that there are other materials included apart from the specimen. There are other materials included, and these materials actually are as follows. We have hand lens. Hand lens will be provided. A red litmus paper and a blue litmus paper will also be involved in the materials that will be produced. And then also we have office pin and tissue paper. Now, as we look at these specimens, you're going to see reasons why the red litmus or the litmus paper is very much important, the office pin and the tissue papers, why they are actually involved, okay? Because they will be also involved in the practicals. Now, the next thing I want to talk to you about before we start looking at each of these specimens and things you should take note of in answering these questions, I want you to note the first thing that is actually important, and that is answering how to answer biology practical questions. How to answer or prepare yourself, let me put it that way, how to actually prepare yourself for biology practical questions or exams. Now, the first thing you should do whenever you are going for any practical exam is to ensure that you enter the exam hall with your writing materials. Enter the exam hall with your writing materials. I've seen several students while in the hall without a complete writing materials, they become destabilized. It really affects their ability to focus on what is on ground or on that part, or to focus in or on the practicals. So one of the things or the materials, writing materials you should come with, one of them is sharpened drawing pencil. Please go with your pencil. Apart from your biro, go with your pencil. And the pencil is preferably 2B pencil, not HB. Let it be a 2B pencil. Number two thing you should also go to the exam hall is your eraser and also your ruler. And I'm going to explain why you need the ruler. The ruler is mostly for your guide, for your labeled lines. It must be all horizontal and ruled. We're going to be talking about that. Now, another thing again you need to do to prepare yourself for biology practical questions or exams is to properly observe any specimen that is given to you. Please take your time to observe them. Sometimes most of these specimens, some of the questions can come based on structural adaptive features. Some of them might come from observable features, features you can observe. So it is important that you properly observe. You take your time to observe every specimen that is being provided for you. And another thing again is that your spelling must be correct to score. So in other words, if your spelling is not correct, you will not be given the mark. Okay, so try as much as possible to work on your spellings. Work on your spellings. Mostly the specimen that will be provided for you, please work on your spellings. Learn to spell them correctly. Learn to spell them correctly. So spelling must be correct to score a mark. Number four important thing you should take note of when preparing for any biology exam or practical exam is that you should read the instructions and questions carefully before answering it. Read it carefully. Now, sometimes in biology practicals, they might say with an annotated diagram. Now, an annotated diagram is quite different from a general diagram. Annotated diagram, you need to finish drawing. After drawing, you give a little explanation of what you have just drawn. I'll give you an instance. You might be asked to make an annotated diagram of the sperm cell. Now, the sperm cell, all you just need to do is, first of all, draw the labeled diagram of the sperm cell, and then from each labels, you can pick out functions of those parts. All right. For instance, we have the head, which consists of the acrosome and the nucleus. You tell us what they do. You talk about the middle piece, which consists of the mitochondria. You tell also what it does, and then the tail that helps to propel the sperm cell forward. So this is what we call an annotated what? Diagram. Now, when they tell you to state, please state. When they say mention, please mention. When they say define, go ahead and do your definition. If they say describe, it is quite different from definition. So please try as much as possible to read the instructions and read the instructions and the questions carefully before you answer them. Another important thing, again, you should take note of is your drawing. In biology questions or practical questions, drawing is paramount. It's so important. You learn how to draw. You learn how to draw. Now, when drawing, please take note of the following. Number one, take note of the title. Your title is important. Every time you are drawing in biology, and of course, expect diagrams, not just one, expect diagrams in these practicals. It will come. It is for sure. It's always compulsory. You see diagrams in biology practicals. So please take note of your title. Let your title come immediately after the diagram. And when drawing your title, if there is a view you are actually drawing with, let the view be added to the title. You can say a lateral view of a tilapia fish. Okay, that is a better title to give. Please don't say specimen A or specimen B or say diagram of specimen A. Please try as much as possible to write the name of that particular specimen. Another thing, again, you should take note while drawing is the magnification, because they won't ask you, but these are the areas where the who will I call them, those that mark your results or your questions, they look out for. They look out for these things. So you write out your magnification alongside with your title. Your magnification must be there alongside with your title. Now, how do you calculate your magnification? All you need to do is check the size of your drawing and check the size of the specimen that was provided for you. So magnification can be calculated as size of drawing all divided by size of the object. Size of drawing divided by the size of the object. Also, another important thing to take note of when drawing is the size. Most times in biology practicals, they will tell you, draw a labeled diagram 6 to 8 cm. Please take note of it. Don't wave it off. Please take note of it. What they want you to do is that your diagram should not be below 6 cm, or your diagram should not be above 8 cm. That is when they say 6 to 8 cm. Okay? Sometimes you even see them tell you 10 cm to 12 cm. Please, when they say 10 to 12 cm, it simply means your diagram should not be below, that's the size of your diagram, should not be below 10 cm. It should not be above 12 cm. So please take note, be careful of the size. Another important thing, again, to take note of when drawing in biology practical is the clarity of lines. Your lines must be clear and not woolly. It must not be woolly. And please try as much as possible to avoid shading of diagrams in biology practicals. Don't shade, don't color. Don't shade and don't color. And that's why we asked that it is preferably you use 2B pencil. Now, your lines must be clear. It must not be faint. It must also not be woolly. It should always be clear. Another thing, again, is neatness of labeled lines. Now, your labeled lines must all be horizontal. It should not be diagonal. It should not be vertical. Please, all the lines must be horizontal on both sides. If you're making labeled lines on the right and also on the left, they all must be horizontal. And I told you about coming to the exam hall with your ruler. They must all be ruled. You must observe this, or else you don't get the score for your diagram. No matter how neat, no matter how beautiful your diagrams or your diagram is, if it does not follow these tips I'm giving to you, you will not get the full mark of your diagram. So please take note of the title, take note of magnification, take note of the size, take note of the clarity of lines, take note of neatness of lines. All must be horizontal and ruled. And then also take note of your labels. Please, when labeling, ensure you label key parts. Label key parts. Label key parts. For instance, if I'm labeling a fish, the key parts are the fins, are the gills, which are the upper column. You have the mouth, or you have the eyes, and so on. Please take note of key parts and label them accordingly. Label them accordingly. And please, when labeling, also let your labeled lines or your guidelines or labeled lines touch what the particular part of the specimen you want to label. Let it touch it, so they can actually understand that this is what you are trying to label or identify in your diagram. Now let's take a look at a format of a good labeled diagram. Now you can see that. Now you see that the neatness of lines are all horizontal. They are all horizontal. You see that the lines are not woolly, okay? They are not woolly. And if you're doing any section in that part where you actually did a cut or a section, you must draw it. When drawing it, it must be with double lines. It must be with double lines. So I believe if you follow these principles or you follow these little tips, and I know and I believe you're going to do well in your biology practical exam. Now let's quickly move ahead and look at several things about the specimen. Now specimen A, we said, is the gills of the fish. Gills of the fish. Now let's talk a little look at some of the areas you should take note when looking at the gills of the fish. One of the things you should actually look at is take note of the diagrams of the gills of the fish. Very important. Take note of the diagrams of the gills of the fish. I have explained to you what the diagrams are, or how you should actually put your diagrams on paper. You should ensure that it is accurate. Follow that principle or the tips I actually gave. Your title, your magnification, your size, clarity of lines. Take note of the neatness of labels, and then your labeling. These are the key areas that will actually award you the mark you actually deserve. Another important thing again to note is to take note of the functions of those labeled lines. Now, if you look at the structure of the gills of a fish, you will discover that the gills of a fish, actually, there are three important labelings you should not forget, you should never miss. You should never miss. And these are the gill filament, we have the gill arc, and then we have the gill rakers. So please also take note of their function. So when labeling, I expect, or you are expected to label the gill filaments, label the gill arc, label the gill rakers. Okay? These are the areas you are expected to make labeled lines or to label accurately. So what is the function of the gill filament? Now the gill filament is the part of the gills where gaseous exchange actually takes place. That is where gaseous exchange takes place. Now the gill arc is actually the foundation on which the gill filament is built. So it actually the basement or a base through which you get the gill filament is actually built. And then we also have the gill rakers. Now the gill rakers helps to stop food from entering into the gill chamber. Now most times you should note the working operation of the gills of the fish, how the fish uses the gills when in water. Now when in water, a fish, when it wants to take in oxygen, that is like breathing in and out, what it does is that it opens its mouth and closes the upper column, so water rushes from the water bodies into the mouth of the fish. Now, as it is entering into the mouth of the fish, it gets to the gill chamber. Now, at the gill chamber, the gill rakers helps to prevent food particles from entering into the gill chamber, while when the water gets to the gill filament, gaseous exchange actually takes place. Now, that means that oxygen diffuses into the blood, and then carbon dioxide from the blood diffuses back into the water. So after this gaseous exchange, the fish closes the mouth and opens the upper column for water that is actually rich in carbon dioxide to come out of the fish. So this is how the fish actually carries out breathing in and out. Okay, so please take note of this. Now, let me show you one or two things to also take note of in terms of the fish, the gills of the fish. Note that the gills is found in aquatic organisms, okay, in organisms that live in aquatic habitats. Also, please take note of the common features of specimen A, which is the gills of the fish, and the next one we're going to be looking at. So there is actually a big similarity, and sometimes you might be asked to write similarities between two specimens. But as we continue, we're going to be looking at those aspects. Now, but let's take a look at the structure of the gills of the fish. The structure of the gills of the fish. Now you can see with me, that is the diagram of the gills of the fish. So you see the gill filament, you can also see the gill rakers, you can also see the gill arc. So please take note while drawing. As you can see also in the drawing, the lines are all horizontal. They are all horizontal, no vertical lines. And you can see that the guidelines or the labeled lines are actually touching the part that is to be labeled. They are touching the part that is to be labeled. And please do not use arrows when labeling, okay? Please avoid using arrows on the labeled lines. So you can do it this way. It is very, very okay. Now, the next one is specimen B. Now, specimen B is whole liver from domestic fowl. The whole liver from domestic fowl with gallbladder attached to it. That is, the gallbladder is attached to the liver. Now, it is also important to take note of some important areas. Now, one of the things I want you to take note of based on my own exposure, I feel questions will come on functions of the liver. So please take note of the functions of the liver. Now, the liver carries out several functions, because what we're looking at here is not just only the liver, we're looking at the liver and the gallbladder. Now, the liver, when it is associated with the gallbladder, plays a major role in digestion. You know, the liver is actually, and the pancreas are actually connected to the duodenum, and they play a major role even in humans, all right? And also they play a major role in other smaller animals or mammals. And we're actually looking, you should also, like I said, take note of the functions of the liver. One of the functions of the liver is in regulation of blood sugar. The liver helps to regulate blood sugar alongside with a hormone which is called the insulin and the glucagon. The insulin helps it to convert excess blood sugar into glycogen, and while the glucagon helps also to convert glycogen back into glucose. So the liver plays a major role in regulation of blood sugar. The liver also plays a major role in detoxification. So when there are poisonous substances that enters into the body through food or through ingestion of food and all the others, the liver handles that matter by detoxifying every toxic substance that enters the body. all right? So the liver carries out the function of detoxification. Another major function of the liver is in the production of heat. Now, the activities of the liver, in fact, the liver is one of the organs that are always that is so active. So most of the activities of the liver actually makes the liver produce what? Heat. So great amount of heat is actually produced from the liver that helps in keeping the body warm. Also, the liver is a reservoir of blood. It's a reservoir of blood. The liver also helps to regulate fats and oils, and I'm going to be talking about that later. But also, the liver helps to regulate blood proteins. And how does it regulate blood proteins? The end product of protein is amino acid. So the liver carries out a process we call deamination, that is breaking down of excess amino acids into harmless substances such as urea, uric acids, and so on, which are later excreted from the body through excretory organs such as the kidney, we have the skin, and so on. Okay, so this is also another important function of the liver. Now, the liver, like I said again, helps also to regulate fats and oils. That simply means that the liver produces a substance we call bile. A substance we call bile. Now, if you have ever dressed a chicken, you're going to have you must have noticed the bile. When you are actually removing the intestine of the chicken, you are going to stumble on the bile. It is actually a kind of a green knob that is actually attached to the intestine. And much when I was actually growing up, we were told that if it bursts and touches any part of the meat or the chicken or that part of the meat becomes bitter. And that is actually the bile. But what is the major function of the bile? Now, when the liver produces this bile is stored temporarily in the gallbladder. Hence, the specimen we have. We said that specimen is a liver attached with the gallbladder. So the gallbladder is intact. So when the liver produces bile, the gallbladder stores the bile. Now, what is the function of the bile? The bile helps in what we call emulsification of fats. Bile helps in the emulsification of fats. Emulsification of fats. And what is emulsification of fats? It is actually the breaking down of fats into tiny droplets. So if you look at the diagram again, you see that you can see the liver, you see the gallbladder beneath the liver, green in color. The liver is actually red, as you can see here, and then beneath it is the gallbladder. Like I said before, the gallbladder stores the liver. It actually stores the liver stores the bile rather, when the liver actually produces the bile. All right, and you can see that it is connected to the duodenum, okay, where it brings about the emulsification of fats. And I've said that emulsification simply means breaking down of fats into tiny droplets. So it brings about emulsification of fats. Now, moving ahead, the next thing I want you to take note of the structure attached to the liver, that is the gallbladder, and their functions. I have told you the functions of the gallbladder, that it carries out the function of storing the bile, okay, the bile. And then you also may be asked to use an office pin. Please note this. Remember, if you go back where we were looking at the specimen that will be given to you. I also told you of other materials that will be needed or will be provided. I mentioned the hand lens, I mentioned litmus papers, I also mentioned office pin and tissue papers. But please take note, the office pin and the litmus paper will be used in testing the gallbladder. Mark me, quote me right. Please note this. In fact, I am so confident that this particular question will come out in this Waec. I am so confident. They will ask you, that's the reason for the litmus paper. they will ask you to make or pierce the gallbladder. Since it is storing the bile, you pierce the gallbladder to release the bile, okay? And then you look at what they said. They said you may be asked to use the office pin provided for the practical exam to pierce the gallbladder, and using a litmus paper, test the content that oozes out. Now, the content that actually oozes out is the bile. So you use a litmus paper to actually test it. Now, if it is a red litmus paper, it will turn blue. It will turn blue. Now, when a red litmus paper turns blue, it simply means that that particular substance or content is an alkaline. Okay, is alkaline. So please take note. The bile is alkaline. Bile is alkaline. And that is why, you know, during digestion, the food enters, and when the food gets into the stomach, hydrochloric acid acts on it. Now, the hydrochloric acid also provides an environment for which pepsin and renin, which are enzymes in the stomach, acts. Okay, it creates an environment for them to what? Act. Because I think we already know, we should know this, that enzymes work best at specific pH. So if they work best at a specific pH, it simply means that pepsin and renin works best at a particular pH. So from the stomach, the food enters into the duodenum, or while it is getting into the duodenum, the liver secretes the bile. So the bile actually terminates or brings an end to the stomach acid that is actually coming into the duodenum. All right. So the bile is an alkaline. Please take note of this. It's an alkaline. Now also take note of this. Take note of the functions of the bile. Apart from emulsification of fats, the bile also carries out other functions. One of them is that it neutralizes stomach acid. Remember, it neutralizes stomach acid. I told you it brings an end to it. So it neutralizes stomach acid. Number 3 function of the bile is that it helps intestine absorb fatty acids and glycerol, as well as fat-soluble vitamins. And of course, the fat-soluble vitamins are vitamin A, D, E, and K. Right? So these are the functions of the bile you should take note of. Right? Another also important thing I believe you should actually look at is some of the diseases of the liver. Now, we have several diseases of the liver. We have liver cirrhosis, we have diabetes, we have mellitus, we have the gallstone, we have cancer of the liver, we have the infective hepatitis, and so on. These are some of the diseases of the liver. You might be asked to mention 2 or 3 diseases that are associated with the liver. So take note of them: the liver cirrhosis, and we have diabetes mellitus, gallstone, we have cancer of the liver. Cancer actually is an abnormal growth in the liver, in the cells of the liver. We also have the infective hepatitis. So these are some of the diseases of the liver. Now moving ahead to specimen C. Now, specimen C is actually lungs from a domestic fowl. Lungs from a domestic fowl. Now, hearing the word lungs, I believe you should be able to understand that it has to do with respiration. Now, can you also take your mind back? What other specimen have we discussed that has to do with respiration? And that is the gills. So the gills is associated with respiration. The lungs is also associated with respiration. So most times when you see questions in this format or specimens in this format, there is every likelihood for them to ask you similarities or differences between the two of them. So as we go on, you're going to see several things I would want you to take note of. Now, for number 1, take note of the location of the lungs in mammals is at the thoracic cavity. The lungs is located at the thoracic cavity. Now, one of the major functions of the lungs is that it brings about what? Gaseous exchange. It brings about gaseous exchange. Another thing again to take note of the lungs from a domestic fowl is the diagram. Now, take a look at it there. We have the glottis. Then from the glottis, the food or air can pass through the larynx, which is the voice box. From the larynx, it enters the trachea, which is the windpipe. And from the trachea, it enters the bronchus. Now, from the bronchus, it enters into the lungs. Now, inside of the lungs, we have the bronchioles and the alveoli. Now, the alveoli is an air sac, and from this air sac, that is where gaseous exchange actually takes place. Gaseous exchange, that means carbon dioxide diffuses from the blood into the lungs or into the air sac, and while oxygen from the lungs diffuses into the blood streams due to the strong affinity from hemoglobin. Okay, so gaseous exchange actually takes place in the lungs. Gaseous exchange takes place in the lungs. Now, another important thing, again, I want you to take note of is common similarities or common features or similar features between the gills of the fish and the lungs. Remember, the gills of the fish is specimen A, while the lungs is actually specimen C. Now, I believe if you check, there was a little error where I made. I said take note of the common features between specimen A and B. It is supposed to be common features between specimen A and C. Now, let me highlight at least 4 common features between specimen A and specimen C, and that is between the gills of the fish and the lungs of, the gills of the fish and the lungs of domestic fowl. There are similarities. One of the similarity is they both have good supply of blood capillaries. That means they are well vascularized. They have good supply of blood capillaries. Now, also both the gills and the fish and the lungs also have large surface area for high rate of gaseous exchange. That is large surface area, like in the lungs, we have large surface area for high rate of gaseous exchange. The same thing also happens in terms of the gills of the fish. Number 3 important or common features between the gills and the lungs is that they both have thin walls, or they are both thin walled to shorten diffusion distance. They are both thin walled to shorten diffusion distance and make diffusion easier and faster. They make that diffusion easier and faster. And then number 4, the fourth common features between the gills and the lungs is that both of them are permeable to allow gas pass in and out easily. They are permeable. They allow gases to pass in and out easily. So these are some common features between specimen A and specimen C. So please take note of this. I'm also vouching that I believe this particular question will also come out. Common features between the gills of the fish and the lungs. Next is specimen D. Specimen D. Now, specimen D is whole gizzard from domestic fowl. Whole gizzard from domestic fowl. Now, also, if you check specimen B, which talks about the liver and the gallbladder, and I told you it is related to digestion, the same thing in specimen D, there is also a relationship towards what? Digestion. Now, the gizzard is the part of the alimentary canal of a bird. Now, after the esophagus, you get the crop, and after the crop, I think you have the proventriculus. After the proventriculus, that's when you have the gizzard. Now, the crop actually helps to store food temporarily. The proventriculus, also known as the glandular stomach, secretes digestive enzymes that helps in digestion of food. And then we have the gizzard after the proventriculus. Now, the major function of the gizzard is actually for grinding food. And that is why if you look at the gizzard, if you open up a gizzard after killing the chicken and getting the gizzard out, before the gizzard is actually dressed and cleaned, you will discover stones or grits within the gizzards. Now, those stones or grits within the gizzard helps in grinding food. And that is why the gizzard is referred to as a glandular stomach. Please don't mix this. The proventriculus is referred to as the glandular stomach, while the gizzard is referred to as the glandular stomach. Now, after the gizzard, you then have the duodenum and then the intestine. So please take note of it. When you see the gizzard, please note what it looks like. So you can see it on the screen there. We have the gizzard. This is when it is actually cleaned and made okay. But when you open it, in the first case, before cleaning it up to this extent, you will discover that it has grits or stones inside. And I've told you the functions of that. It helps for grinding of the food. We move ahead to specimen E. Specimen E. Now, specimen E is actually a whole intestine from a domestic fowl. The whole intestine from the domestic fowl. Remember, we said that after the gizzard, you have the duodenum. After the duodenum, we then have the intestine or the small intestine, where absorption of food takes place. So please take note of the functions of the intestine. And like I said before, the intestine carries out two major functions. I'm talking about the ileum. It carries out two major functions, mostly in humans. And the two major functions are, number one, digestion is completed in the small intestine. And then number two is that absorption takes place. And absorption simply means the diffusion of digested food from the intestine from the small intestine to the bloodstreams through a finger-like structure we call the villi. Now, I'm describing this based on humans. But please also take note, both in domestic fowl, digestion is completed at the small intestine, and absorption also takes place at the small intestine. Now you can see a diagram of the intestine of domestic fowl. You can see there we have the gizzard. You can see the liver, okay? The liver very close to the gizzard. You can see the crop there labeled. You can also see underneath the liver, we have the gallbladder that is attached to the liver at point of pen. You can see it there beneath where you have this arrow pointing at it. You then also have the small intestine with the pancreas. So this is the pancreas. We then also have you have the duodenum, where this is the duodenum, and then this is the small intestine where digestion is actually completed and absorption takes place. Digestion completed, absorption takes place. Now the next one is specimen F. Specimen F. Now specimen F is ginger rhizome. Specimen F is ginger rhizome. Now ginger is an underground stem. Please note this. Ginger is an underground stem, and it reproduces naturally by use of modified underground stem. You know, in terms of vegetative reproduction, we have two types. We have the natural vegetative reproduction, which has to do with use of modified underground stem and use of fleshy leaves. These are the two natural type of vegetative reproduction. I repeat again, I said we have two types of vegetative reproduction. We have natural vegetative reproduction and we have artificial vegetative reproduction. For natural vegetative reproduction, we have two types again, which is use of modified underground stem. That's where the ginger falls in, and ginger is referred to as a rhizome. We have the suckers, which is the banana. We have the culm, which is the cocoyam. We have so many of them, the bulb, which is the onion, and so on. These are all under use of modified underground stem. We also have the use of fleshy leaves. An example is the bryophyllum, which we'll be discussing much later. And also take note of the part of the plant to which the specimen belong. Now, what part of the plant does a ginger belong to? And that is the stem. Please note it. The ginger is a stem. All right. Now, also take note of the method of propagation of the specimen. Now, it is propagated by cutting the stem. Ginger is propagated by cutting the stem. It is simply because of the presence of lateral buds. Now, let's take a look at other things to note. Now, also take note of some observable features. Remember, I told you whenever you are in the exam hall and you are provided with specimen, please try as much as possible to carefully observe the specimen. Observe the specimen for features. Questions will be asked on the features, observable features. Now, what are some of the observable features in the ginger? The ginger possesses what we call internode and the node. If you have a ginger plant or a ginger stem, you will discover that there are several things that protrudes out. We're going to be seeing it in the picture. That is the node, and then before it we have the internode. The ginger also possesses what we call terminal bud and auxiliary buds. It has terminal bud and auxiliary bud. Now you can see the ginger plant. You can see that these are the buds. You can see the node. The internode is there. You can see that it has a scaly leaf. And then you can also see the adventitious roots that are found with the liver. So it has adventitious roots. All right. So please take note of the parts. You might also still be asked to draw the ginger. You might be asked to draw it. So please take note of it. And remember the tips I told you about while drawing: your title, your magnification, your size, the clarity of lines, neatness of labels, and then your labeling. All right. So take note of all of this. I pray you start. If I were you, I'll start practicing almost all diagrams. Practice all the diagrams so that you can, you will be sure of what to do when it comes. The next one is specimen J. Specimen J. Now, specimen J is actually onion bulb. The onion bulb. Now, in this particular specimen, take note of the part of the plant to which the specimen belongs. Now, onion bulb belongs to a leaf, is a leaf. Onion bulb is a leaf. Ginger is a stem. Onion bulb is a leaf. Ginger is a stem. Also take note of some observable features in an onion bulb. One of them is that it possesses short and compressed stem. You're going to see it in the diagram, short and compressed stem. Now that short and compressed stem most times in some diagrams is called the basal. All right. Now also another thing again you find in an onion bulb is the presence of adventitious roots. So there are presences of adventitious roots. Also, they also possess succulent scale leaf or scaly leaves. They possess scaly leaves, but inside of those scaly leaves, we have a succulent leaf. That is, it is filled with food, so it stores actually food. All right. Now, take note of the mode of vegetation, vegetative reproduction, that is in onion bulb, and it is natural vegetative reproduction by use of modified what? Stem. By use of modified stem. Now look at this. Take note also of some differences between the ginger and the onion bulb. Several times in some past questions, we have seen them asking questions of the differences between ginger and onion bulb. Now, in terms of ginger, in ginger, Node and internode are both present. And then here in onion, node and internode are both absent. We're going to be seeing that. Node and internode are both absent. Also in ginger, there is absence of scaly leaves. What we have is scaly stems. Then also in onion bulb, there is presence of scaly leaves. Number 3, ginger leaves are not fleshy. But in onions, the leaves are fleshy. Also take note of some similarities between the ginger and the onions. Now, they both have or possess adventitious roots, and they also both possess terminal buds and auxiliary buds. So in onion, you have terminal and auxiliary buds, and also you have seen as it relates to ginger that they have adventitious roots, and we mentioned in the features, observable features of onion, we said they also possess adventitious roots. Now let's take a look at the structure of the onion bulb. You can see the adventitious roots. You can see the basal plate, and that is this can be likened to the stem. Then we have the scaly leaves. Now you can see these other leaves, they are stored, they are succulent, all right? Then we also have sprout leaf with what? Blade. So leaves are actually sprouting out. So this is actually the structure of the onion bulb, structure of the onion bulb. Moving ahead, specimen H. Now specimen H is actually leaf of bryophyllum with adventitious roots. The leaf of a bryophyllum. Now, bryophyllum are actually leaves, all right? So the part of the plant of bryophyllum is a leaf. Bryophyllum is a leaf. Now, also take note of some of the features of the bryophyllum. Now, the bryophyllum possess succulent thick blades. Succulent thick blades. They are also blunt at the tip. They also have what we call dented margins. That is, the margins are dented. They are not so straight. So they have dented margins. Another thing again, it is used for propagation. That is biological significance of the bryophyllum. Now, another important thing again I want to mention is that the bryophyllum also possesses adventitious roots. It possesses adventitious roots. So please take note, you can add that to some of the features of the bryophyllum. It possesses adventitious roots. And then let's look at biological significance of bryophyllum. Bryophyllum is used for propagation. It can be used to grow another organism or another bryophyllum. Then number 2, it carries out photosynthesis. Now, the reason why it carries out photosynthesis is because it is actually a green plant. So it possesses chlorophyll, it possesses chloroplast. So these are some of the biological significance of the bryophyllum. Now take a look at the structure of the bryophyllum. You can see beneath these young plants that are developing from the adventitious buds, you will see that they possess adventitious what? Roots. You can see here a bud which will give rise to a young plant is actually developing. So this is the structure of the bryophyllum, the structure of the bryophyllum. Next is specimen J. Specimen J. Now, specimen J is cassava stem. The cassava stem. Now, please also take note of the diagram of the cassava stem. Take note of the diagram of the cassava stem. Remember the tips again on diagrams. Also take note of the mode of reproduction. The mode of reproduction. For cassava stem, they are propagated artificially by what we call stem cutting. So by stem cutting, all right? You cut the stem, and then you can put the stem in a fertile soil, and then it will germinate or grow into a new plant or a new cassava plant, all right? Now, take also note of some observable features. Number 1, in a cassava stem, there is the presence of a bud. there is presence of a bud, there is presence of node and internode. There is also the bud that is present there is not covered with scaly leaves, and then cassava stem does not store food. Cassava stem does not store food. Now, this can also be used as differences between, the cassava stem can also be used as differences comparing it with the ginger. Remember we said ginger is a stem, but ginger actually stores food, while the cassava stem does not store what? Food. Now, these can also serve as differences between the cassava stem and the cassava tuber. But for me, I believe they will not be asking questions on differences between the cassava stem and the tuber. I suspect that they will be asking questions between cassava stem and the ginger, okay, the ginger rhizome, because both of them are similar somehow. They are both what? Stems, okay? They are both stems. Now, take a look at the diagram of the cassava stem. As you can see there, you see the auxiliary bud, all right? We also have the node. You can also see the internode. The node is actually the part that is very close to the bud. And then we also have the leaf scar and so many other things you can see. Now, you see when it is planted, the node or the bud actually starts producing adventitious roots. And then it germinates and grows into a new cassava plant. All right? So this is the diagram. Please try as much as possible again to take note of these diagrams. You can't tell where questions on diagrams will come up, or you can't tell the particular specimen that will give rise to a diagram, or you'll be asked to draw. So please take note of it. All right? Next is specimen K, and specimen K is cassava tuber. Cassava tuber. Now, cassava tuber, take note of the agricultural classification of it. Agricultural classification, that's cassava tuber, it is a stem tuber. Stem tuber, all right? And then also the life cycle is biennial. Now, the reason we say biennial is because it completes its life cycle within 2 years. Please also note that cassava is actually rich in starch, and it's a source of carbohydrate. It's rich in carbohydrate, and also is a source of energy. Now, that brings us to the economic importance of value of cassava tuber. Economic importance. Number one, it is an energy-giving food. So it provides energy for consumption. When consumed, rather, it provides energy for both plants, for both man and animals. Also, it can be used in the industries. That is when we were looking at the processing of the cassava tuber. It can be processed into gari, okay? It can also be processed into, in native languages, they call it fufu, and so on. These are several ways that the cassava tuber can be processed. And that is simply means that it is also a raw materials used in so many industries. And then also it can also serve as a source of income. Many persons have made their money in the sales of cassava, or the sales of the cassava process, or the finished product, processing product of cassava, which is gari. Many persons have made serious money from that. So it can also serve as a source of income. It can also serve as a source of employment. Now, most persons are employed in the farm. You know, as a farmer, that is his job. Okay, one can also employ someone to help in the harvest or the planting of cassava tubers. So it can also serve as a source of what? Employment. And also it can also serve as a source of food. Like I said, it is an energy-giving food. So these are some of this economic importance or nutritive value of cassava tuber. Now take a look at the diagram there. That is cassava tuber. So it also has adventitious roots. You see why we call it a stem tuber? It has stems connected to it. Then you see the root tuber there, and then the stem protruding out from the soil level. So that is a root, a cassava tuber, which is also referred to as, can be referred to as a root tuber. It can also be referred to as a stem tuber. All right, specimen L is thoracic vertebra. Thoracic vertebra. Now, the thoracic vertebra, let me say the vertebral column consists of vertebrae, okay? Now, in man, we have about 33 vertebrae. Now, those 33 vertebrae, they are divided into 5 major groups. We have the cervical, which is located at the neck, and there are about 7 in number. We have the thoracic, which is located at the chest region. I'm mostly coming to it. And there are about 12 in number. We also have the lumbar, which is number 3. It is located at the upper abdomen, and there are about 5 in number. We have the sacral, which is located at the lower abdomen, and also 5 in number. And then finally, we have the caudal vertebrae, which is also known as the tailbone, and then they are 4 in number. So we're looking at the thoracic vertebra. The location of the thoracic vertebra is at the chest region. So it's located at the chest region. We also have the number of vertebrae in humans. It's 12, like I said before. Now, please take note of the features, because definitely you might be asked to draw the diagram of a thoracic vertebra. I think about 4 or 3 years ago, this question, this specimen was actually produced, was used, thoracic vertebra and lumbar vertebra, and then diagrams were required, okay? So please take note of the diagram of the thoracic vertebra and the lumbar vertebra. Take note of their diagrams. Now let's look at some of the features of the thoracic vertebra. The thoracic vertebra has a neural spine. A neural spine. Please, please, again, when drawing the diagram of any vertebrae, please, or any vertebra, please, it is important you note there are key labeled parts you should always label. One is the neural spine. We have the neural canal, we have the centrum, we have the transverse process. Very important. And I'm going to be looking at it based on, that's the area I'm going to be talking about based on the features of the thoracic vertebra. Number 1, these features, they have long neural spine that is actually projecting upward and backwards. So you see it as if it is like this. Long neural spine is projecting upwards and then a little bit backwards. So apart from that, number 2 feature of the thoracic vertebra is that it has a pair of short transverse process. So the transverse process, which are usually found at the sides, are usually short. And I'm going to be telling you the functions of the transverse process. We're also going to look at the functions of the neural spine. We're also going to look at the functions of the neural canal, all right? So please take note, the side projections, the side bone projections is the transverse process. In the thoracic vertebra, it is actually shortened, it is short. And then also, thoracic vertebra has large neural canal. Now, the neural canal is actually that opening or hole that is found in any vertebra. And then number 4 is it has a large cylindrical centrum. Now the centrum is a hard piece, is a piece of bone, okay, that is found, a stronger hard piece of bone that is found in the vertebra. Now let's take a look at the functions of the thoracic vertebra. Very important. They might, in fact, they will ask it. Functions of the thoracic vertebra. Number 1, it aids the attachment of the ribs. It aids the attachment of the ribs. Now, if you take a look at the rib cage, the ribs are attached to the sternum and also to what? The thoracic vertebra. The ribs are attached to the sternum and also to the thoracic vertebra. Remember, we have about 12 pairs of ribs, and these 12 pairs of ribs are attached to the to the thoracic vertebra, all right? So that's one of the functions. It aids the attachment of the ribs. Number two, muscles of the shoulder and back are attached to the neural spine. And that brings us, we'll be talking about the functions of the neural spine. One of the major functions of the neural spine is for muscle attachment. So the muscles of the shoulder and the muscles of the back are attached to the what? Neural spine of the thoracic vertebra. Then number three is that it aids in breathing alongside with the ribs. So while you're breathing in and out, the thoracic vertebra plays a major role. Remember, when you're breathing in, the ribs move upward and outward, moves upward and outward. Now, as it is moving upward and outward, the thoracic vertebra is also involved in the movement. So it also helps the ribs to actually achieve that movement. All right. Then we also have the next one. During breathing out, the same thing. The ribs not move upward; it moves downwards and inwards. The same thing too. The thoracic also helps the ribs to move downwards and inwards. All right. Then we also have some important functions of the following. Let's look at the functions of the neurospine, look at the functions of the transverse process, look at the function of the neural canal, and also the centrum. Now, the function of the neural spine is for attachment of muscles. I mentioned that earlier. The neural spine provides attachment of muscles. Then we also have the transverse process. Remember, I told you that the transverse process are the side projections, while the neural spine is actually the upward projection of bone. Then the side projection has to do with the transverse process, and you have two transverse processes. And what is the major function of the transverse process? The major function of the transverse process is that it aids attachment of muscles and ligaments. It aids attachment of muscles and ligaments. We also have the neural canal. The neural canal carries out the function of, it serves as a passage for the spinal cord. Remember that the vertebral column or the backbone actually protects the spinal cord. The spinal cord is actually one of the central nervous system or coordinating system of the body. Very vital and also very, very delicate. So what protects actually the spinal cord is the backbones, and the part through which the spinal cord passes to the brain through the backbone is the neural canal. And then we also have the centrum. Now, the centrum is a solid piece of bone that is actually found below the neural canal. Now, let's take a look at the structure of this vertebral column. Now, take a look at it. Now, you see the sponneous process, which is the same thing as the neural canal. You can see it is projecting upwards. Now, look at how it is in the lateral view. Remember, I said that it projects upwards and backwards. Project upwards and backwards. As you can see through the lateral view, it is projected backwards. You see a large neural centrum, which is called here the corpus, is a large centrum. Then we also have the shortened neural transverse process. You can see them by the sides. All right. So these are all important things to take note of. And then we have the vertebral foramen, which is also known as the neural canal. You see a large neural canal. So this is a diagram of what? The thoracic vertebra. The diagram of the thoracic vertebra. Moving ahead, we have specimen M. Now, specimen M is the humerus bone. Humerus is actually located at the forelimbs. That's the location. Is located at the forelimbs. Now, in the forelimbs, what we have a very large bone or a long bone, which is the humerus, and then after the humerus, you have the bones, two bones after the humerus, two shorter bones, and they are called the radius and the ulna. After the radius and the ulna, you have the wrist bone, which is called the carpals. After the wrist bone, you have the palm bone, which is called the metacarpals, and then you now have the phalanges, which is pentadactyl, simply meaning that they are 5 digits. That's the phalanges. The phalanges are pentadactyl mostly in humans. All right, so this is the 4 limbs. Now, but take note of some of the features of the humerus. Now, the humerus consists of a head. It also has a shaft. Now, note that it has a head. The head is actually round, and it articulates and fits into what we call the glenoid cavity. Now, the glenoid cavity is found in the scapula. So it means that the humerus attaches with the scapula through the glenoid cavity to form what we call the shoulder joint. It forms the shoulder joint. And this brings us to what we call the pectoral girdle. So this is the pectoral girdle. The pectoral girdle consists of the scapula, which is the shoulder blade. It also consists of the clavicle and the glenoid cavity and coracoid. Okay, these are what makes up the pectoral girdle. But the humerus attaches with the glenoid cavity of the scapula to form the ball and socket joint. Remember the ball and socket joint? It allows movement in every direction. One of the examples of the ball and socket joint is the shoulder joint. And the humerus brings about the articulation of the humerus with the scapula brings about the shoulder joint. Another important feature to take note of in the humerus is that it possesses a grooved pulley at the end called the trochlea. Now, this trochlea articulates or fits in with the radius and the ulna to form the hinge joint. Now, the hinge joint allows movement in only one direction, back and forth, back and forth. Now, this the type of hinge joint that is formed by the humerus and the radius and ulna is called the elbow joint. So it forms the elbow joint with the humerus, with the radius and the ulna. And also finally, I said that the humerus possesses a long and strong shaft. It possesses a long and strong shaft. Now also, nutrients that are contained in the humerus should be taken note of. Questions can come up from that aspect to actually mention some of the nutrients that are contained in the humerus. Now, take note of these nutrients. Mostly bones consist of calcium and phosphorus. So the bones are the nutrients or the mineral elements are calcium and phosphorus. Now, these calcium and phosphorus are for strong bones and teeth. That's their function. So they function for strong bones and teeth. Now, let's take a look at the functions of the humerus. The humerus plays a very vital and important function. One of them is that it gives the body rigidity and support. The humerus gives the body rigidity and support. So it provides support for the arm, all right? Number 2, it gives shape to the body. One of the major functions of every bone is that it gives shape to the body. Number 3 is that it provides the surface area for the attachment of the muscles. Remember, muscles are attached to bones through a connective tissue we call the tendons. So the tendons allow or helps for the attachment of muscles to bones. Number 4 function of the humerus is that it articulates with the glenoid cavity of the scapula to form the shoulder joint, and also it articulates with the radius and ulna to form the elbow joint. Okay, so these are some of the functions of the humerus. Now let's take a look at the structure of the humerus. As you can see there, we have the radial groove. Now that radial groove there is the strong and long shaft. As you can see, we have a strong and long shaft. Now you can see the head. This is where what fits or articulates with the glenoid cavity of the scapula. And then here is where we have the olecranon fossa. That is the one that actually articulates with the radius and the ulna to form the elbow joint, all right? So this is diagram of the humerus. Next is specimen N. Now specimen N is mango seed. Mango seed. Now, looking at mango seed, I'm suspecting a question can be asked concerning fruits. You know, the seeds are actually located in a fruit. Now, what is a seed? A seed is a fertilized and developed ovule. A fertilized and well-developed ovule. Please take note of that. While a fruit is a fertilized and developed ovary, okay, of a plant. So the fruit is found within the seed and the embryo is found within the seed, okay? The seed is found within the fruit and the embryo is found within the seed, all right? Now take note of the types of fruit it is gotten from. Now the type mango fruit is actually a drupe. It is actually a drupe. Also, please take note of the features of a drupe. Now, most times drupe, they have thin epicap. They have thin epicap. They have sometimes, will I call it fibrous or fleshy mesocarp? Yes, like in terms of coconut. Coconut has fibrous mesocarp, while mango has fleshy mesocarp. And then their endocarp are usually stone hard, are very stone hard, or you can call them stone hard. They're very, very hard, okay? And they are not edible, all right? So that is in terms of drupe. And note that if they are asking on mango seed, definitely, which is a drupe, definitely there's going to be a specimen that has to do with a berry, all right? So please note similarities and differences between drupes and berries. Now, apart from that, looking at a mango seed, every seed consists of several things. We're going to be seeing that in the diagram. The seed consists of a tester, it consists of a micropyle, it consists of the radicle, the plumule, and then the cotyledon, which also is a form of the embryo. Now, please take note of those areas and what they are for. Now, mango seed, I believe it is a dicotyledonous seed. And since it's a dicotyledonous seed, I believe it has two seed leaves and also undergoes epigeal germination. Mango seed undergoes epigeal germination. Also, please take note of the conditions for germination. There are several conditions that are necessary for germination to take place. The seed must be viable, there must be good temperature, there must be moisture, there must be a good soil must be present, relative humidity must also be okay, enzymes must also play a major role in the germination of the seeds. So these are the things you should take note of on the mango seed. Now, if you look at the structure of the mango seed there, we have the plumule, we have the radicle, we have a micropyle, we also have tester and the cotyledon. They are also found in the mango seed. Now, the next one is specimen P, which is femur bone. Now, the femur bone, remember we just finished talking about the humerus. Now, the femur bone is located at the hind limbs, which is the leg. Now, the hind limbs consist of a long bone, which is the femur, and after that we have the tibia and fibula, which are two bones found at the beneath, immediately after the femur. And then after the femur, after the tibia and fibula, we have the ankle bone, which is called the tarsals, and then we have the feet bone, which is called the metatarsals, and then we have the phalanges, which is actually pentadactyl. That means 5 digits. Now, also, please take note of some certain features of the femur bone. Take note of the features of the femur bone. Number one, the femur bone has a round head, the same way we have the round head in the humerus. So sometimes you might be asked to describe common features okay, observable common features, or you can also be asked sometimes of differences, which we are going to see much later. Now, note that it has a round head, and this round head articulates with what we call the acetabulum. Now, the acetabulum is located at the hip region, that is the hip girdle. Okay, the pelvic girdle rather, that's the pelvic girdle. In the pelvic girdle, we have the acetabulum, where the head of the femur articulates to form the ball and socket joint, which is also known as the hip joint. It forms the hip joint. And the hip joint, or the ball and socket joint, is a joint that allows movement in every direction. Number two feature of the femur is that close to the head of the femur, there are three projections. There are three bone projections. And those three bone projections are called trochanters. Okay, they are called trochanters. Okay. Now, these trochanters actually provide attachment for the muscles. They provide attachment for the muscles. Number three important feature of the femur bone is that it possesses two rounded knobs called condyles, which articulate with the tibia and the fibula to form the what? Hinge joint, which is also known as the knee joint. It forms the knee joint. And there is also a cap that covers the knee joint in front, and that cap is known as the knee cap, or you can call it patella. All right. Then also number four rather, feature of the femur bone is that it possesses a long and strong shaft. And number five, don't forget this, the femur bone is the strongest and longest bone in the body. So the longest and strongest bone in the body is referred to as the femur bone. That is the femur bone. Now, also, let's take a look at nutrients contained in the humerus, in the femur rather. It is also the same as that contained in the humerus. So the femur also has calcium and phosphorus. And this calcium and phosphorus are for strong bones and teeth. Please don't forget. Now, what are the functions of the femur? Number one, it gives the body rigidity and support. Number two, it is used for locomotion. It helps to bring about movement. It helps to bring about movement mostly in animals. Number three, it gives the body shape. So the shape of the laps is actually based on the shape of the femur. And then number four, it provides the surface area for the attachment of muscles. And then finally, number five, it articulates or fits with the acetabulum and the tibia and fibula to form the hip joint and the knee joint respectively. All right. So by this, what it means is that the head of the femur fits in with the acetabulum to form the hip joint, and the end of the femur, which is, I think that's the condyles, they fit in with the tibia and fibula to form what? The knee joint. So these are some of the functions of the femur. Now take a look at the structure of the femur. You can notice the head, okay? You can notice the trochanters, which are the three bones that provides attachment for the muscles. We have them here. Okay, you can also see the neck of the head, and then you can see this long shaft. You can see the condyles. Okay, the condyles provide attachment for the tibia and the fibula. All right. Now, next specimen is specimen Q. Now, specimen Q is lumbar vertebra. Remember, we just finished talking about the thoracic vertebra. Lumbar vertebra is also provided. Now, where is the lumbar vertebra located? It is located at the upper abdomen immediately after the thoracic vertebra, which is located at the chest region. So the lumbar is located at the upper abdomen. Now, in humans, we have about 5 lumbar vertebrae. Excuse me. In humans, we have 5 lumbar vertebrae. Now, what are the features of the lumbar vertebra? Number one, the lumbar vertebra has a broad and flat neural spine. It has a broad and flat neural spine. Number two is that it has a large and flattened transverse process. Large and flattened transverse process. Number three, it has a large neural canal. A large neural canal. And then number four, it has a large and thick centrum. So these are four important features of the lumbar vertebra. Now let's take a look at functions of the lumbar vertebra. Now the lumbar vertebra, number one, it provides attachment for the abdominal muscles, and number two, it bears the considerable weight of the body. It bears considerable weight of the body. Another thing again about the lumbar is the structure. Please look at the diagram. You can see the neural spine is flat and large. All right. You can see the neural canal large. You can see the transverse process well pronounced. All right. You can also see the knobs that comes out close to the transverse process, and that makes it actually different from the thoracic vertebra. You can see the neural spine. See how it is. Okay, it is quite different from the thoracic vertebra. In the thoracic vertebra, we said it is projected upwards and backwards a bit. All right. So these are important areas we should actually take note of when taking the diagram of the lumbar vertebra. Please also try as much as possible to remember the structure of the lumbar vertebra, the diagram, very important. Questions might be asked to actually draw this diagram. So we're going to be looking at the last specimen for the Waec practicals biology, and that is specimen R, and that is orange seed. Remember, we actually discussed the mango seed, and I told you to take note of the fruit from which that seed is actually coming out from, the type of fruit. And you should also do the same for that of orange seed. Now, for orange seed, the type of fruit it is actually a berry. So orange fruit is actually a berry, and this orange fruit has the seed. Now, also, please take note of the functions or features of berries. Now, we said that a berry has thin epicap. It has a fleshy mesocarp, which is always edible. And then also, it has a fleshy endocarp or soft endocarp. All right? So it has a soft endocarp. It is not hard. And then also, if you compare the seeds of mango seed and orange seed, you will discover that mango seed is actually hard compared to that of the orange seed. Also, please take note of the mango seed. Mango seed is also dicotyledonous seed. It has two leaves, seed leaf, and it also undergoes epigeal germination. All right, it undergoes epigeal germination. And also another major difference between berry and drupe is that drupe actually produces one seed that is usually large and stony hard. But in terms of berries produces many seeds, and these many seeds are actually small. But if you look at that of orange, the orange seed, and if you look at the orange and its seed, you will discover that the seeds of orange are arranged in chambers. You'll see that the fruit is actually divided into chambers. That is, if you do a transverse section of the orange fruit, you will see the seeds are arranged in different chambers, and there are more than one. All right. Also, please take note of the conditions that are necessary for germination to actually occur. All right. And I think if you do that, you'll be good to go. So this is the orange seed. Take note of it. I have no clue yet as to some of the questions that will be asked concerning the seeds, but I believe maybe there'll be common features can be actually mentioned. Several things can also be stated that is similar with that of the mango seed, that is actually similar to that of mango seed. You can have also differences and then also similarities. Some of the differences is that they are produced, the seeds in orange seeds are many in their fruits. Mango seed is just one. The orange seeds are not stony hard. Mango seeds are actually stony hard. And there are so many other things to mention looking at both of them. And then also, if you're looking at their similarities, you also discover some of the things there. I think the orange seed has a tester. For mango seed, I'm not too sure. The tester is there, but I think so. And then also they have plumo, they have radical, they also have cotyledons. Since they are dicotyledonous plants, it simply means they have two seed leaves. And they also undergo the same type of germination, which is epigeal germination. So these are the few information I actually have, and I believe it is going to do you well, and I believe it's going to actually help you during your exams. From me, I am actually wishing you good success in your biology practical exam. Bye for now. Have a nice time.